Good morning or good afternoon, everyone. On behalf of the Biochemical Society and Portland Press, I'm pleased to welcome you to today's webinar, which is part of our biochemistry focus webinar series. Today in the series include different research areas in the molecular biosciences as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. My name is Mohammad Awais, and I'm from the University of Liverpool. My research focuses on developing novel therapeutics for acute inflammatory conditions, with a particular focus on acute pancreatitis. Since my PhD at the University of Tokyo back in 2005, microscopy has been at the heart of my work. I have used advanced imaging extensively to investigate dynamic cellular processes at the single cell level, and it continues to be a central tool in driving my research forward. Our goal today is simple: to shape a future where microscopy data is not only high quality, but also reusable, transparent, and trustworthy. Reproducibility isn't just a technical requirement. It is a cornerstone of scientific progress. In today's webinar, we will hear from Dr. Marie Held from the University of Liverpool. During this session, Marie will discuss why reproducibility matters for scientific community and for our own work, and how it aligns with the FAIR principles, which are findable, accessible, interoperable, and usable. Questions will be asked after Marie's presentation, but you can send in your questions during the talk. And if you have a question, please type it in the question box as shown in the image on the screen. And we will try to answer as many as time allows. And over to Marie.
Thank you very much for the introduction. Can you see my slides?
Yes, I can see your
Just as a
slides.
sanity check. Okay.
Yeah.
So as said, my name is Marie Held. I am an image analyst in the Center for Cell Imaging at the University of Liverpool. I have been in position for 6 years by now, nearly. And the presentation I'm giving today is a very condensed amount of information that I have learned along the way over years. So it's got to be a lot of information, but if you can take away maybe the odd practical thing, then I've done my job well today. So disclaimer, first of all, I have based my presentation on a more generic presentation, from a reproducible research workshop, which was licensed under CC BY4 license, and therefore my slides are also under CC BY4 license. What is reproducible research? Well, if we want to go with FAIR, then we need to know what FAIR means. Mohammed has already said so. So findable, accessible, interoperable, and reusable. Reproducible research, whether it's microscopy based or not, there are a lot of things here today that are transferable to other things, is research, whether it's in a scientific or kind of a natural sciences background, or whether it's humanities or arts or other subjects, is research that is sufficiently transparent that someone with a relevant experience can clearly follow as relevant for different types of research. They can follow how it was done, why it was done in that way, in that particular way, the evidence that the research has established, the reasoning and or judgments that were used, and also how all of that led justifiably to the research findings and conclusions drawn from the work. So in 2016, a publication emerged where 1,500 scientists were asked whether they can reproduce data that is in the literature. Over 70% of these researchers have tried to replicate another researcher's experiments and failed. Over half had failed to reproduce their very own experiments, which is not good. Half of the researchers agreed that yes, there is a significant crisis on reproducibility. This has a manifold number of reasons. There is certainly difficulty in managing complex data sets. There is poor statistical processes, poor research design, including failure to control for bias, a lack of access or detail of the methodology used, a lack of access to the raw data and research materials. We all live in a publish or perish culture that only warrants novel findings, not negative findings, and only kind of sexy findings. And however, what each and every one of us can do is to focus on making small improvements to our very own practice and advocate for wider improvements, as kind of trying to get a culture change going. There are different approaches that you can use that go throughout the whole process of a research project. So from pre-registration of experiments to more training on study design and statistics to publishing of preprints, more support for publishing negative results, but also more replication and validation studies conducted and shared. If that hasn't convinced you, maybe I'll appeal to everybody's selfish reasons to work reproducibly. So reproducibility helps to avoid disaster. It makes it easier to write papers, and who doesn't want to have an easier time writing papers? It helps the reviewers see it your way. That means you can publish your paper, hopefully, slightly easier. It enables continuity of your work and also helps to build your reputation. So working reproducibly has many benefits for the research ecosystem and as a wider area, but also direct benefits for yourself. So I've kind of, the presentation is organized as 7 different steps, which are kind of from start to finish of a research project. First of all, let's plan to be organized. If you're going down the PI route or you are a PI, then you definitely have heard the words data management plan. To have a data management plan sometimes seems like a box ticking exercise. However, there are benefits to be harnessed here. A data management plan is a living document for a research project. It doesn't lock you in, it's not a prison. But what it does outline is data creation, what data is created and how, using which equipment, data policies, how long are you storing your data, where are you storing your data, how do you access your data, access and ownership rules, management practices, management facilities and equipment, and who will be responsible for what. You don't want to be in the situation like I was. The group I did my PhD in, the PhD supervisor was always, you keep the data, and if I need the data, I'll send you an email in like 10 years time, 15 years time. That's not a great plan. Second thing to do is keeping files tidy and organized. What I've done in the past was organize folders by date, which is an organization method. However, there's a better way of going about it. So keeping a folder structure where everything relative to the project, whether it's a microscopy project or a different project that has microscopy in it, and have all the relevant data available within that. So you have your sample preparation. This will be which chemicals, which consumables did you use, which cell lines if you're doing microbiology, which for microscopy, which antibody staining or dyes did you use, fluorescent dyes. Then you might be doing some imaging. This is where you store your imaging data, but also your metadata for the imaging. Which microscope did you use? Which objective? What are the imaging parameters that you applied? Maybe you use different microscopes. Which one has taken, or on which microscope have you taken which images? If you do Western blots and you take an image of that, this can all be collated in here. If you're doing image analysis, you want to save your script, intermediate files, your results files in here. And then you also have downstream data analysis. And for each project, what is a very good idea is to have a README file. This is a reminder for you and for other people where you can say, what is where? You can have here a list of, these are the pieces of equipment I've used without the ultimate details, or I have processed so many samples, where is the data saved? Anything that pertains, where did I get the cell line from? If you're collaborating with somebody else, oh, so-and-so sent me the cell line or the dye or something else. So this is all information that you can collect in here. Also, important discussions, email threads, is like, oh, check that email from that day. This is all important information pertaining to the microscope or the project as a whole. Okay. Yes, I have said all of that. I'm not showing you these bits. Another key part is file naming. This is something as working in a facility that I see a lot where people make their own life much harder than it needs to be. And there are actually 3 key principles of file naming, which are simple to remember and to implement. Make the data or the file names machine readable. Use only letters, numbers, dashes, and underscores. Only because the operating system allows you to use percentages, brackets, special characters, doesn't mean it's a good idea, spaces, to do so. Case sensitivity is your friend to convey information. Also make it human readable. Use descriptive words that make sense. Maybe a cell line HeLa, or maybe if you're doing a time series, this is day 7, D7, or something else that makes sense to you. So that when you look at the file name, you already have a good idea of what is in that file. And you want to follow a convention that plays well with default ordering. If you're taking 100 images, don't start image 1, 2, 3. Start with image 001, 002, 003, because then your images are going to be in nice chronological order. Also, use a date format that has the year, ideally 4 spaces for the year, then the month, then the day. If you have this as the first part of your file name, all your files will be in chronological order like this. So it's much easier to identify what is the most recent file. You don't have to look at the date modified or date created column. You can actually see it from the file name. So if you had to submit a report, would you rather look through this list of reports or this list of reports? Here it's really easy. The last one is most likely the most up to date. So this one's the one to submit. Make your life easy. And also, if anybody else has to look at your data, they can have a much easier time to identify what's what. Ensure that things like data sharing are considered throughout, and also it might be recommended but not mandated by your institution to use a certain file naming convention, so do check with your library. Another concept that I would like to introduce is the concept of tidy data. This is not something that comes naturally, but it is really useful in the long run. So what is tidy data? It fulfills three criteria. Each variable must have its own column. Each observation must have its own row, and each value has its own cell in a table. So if we're looking at this table, this is not tidy data. This is, I guess, messy data or wide data. Why is it messy? Well, these are the treatments. These are, in fact, variables. These things are also variables because you have the concentration of the various treatments. And then here we have multiple observations per row. Whilst this is the standard way that people create tables, and sometimes it's also a quicker way to plot something in Excel, for example, it's not a good long-term solution, because also, what do these numbers stand for? What was measured? That information is not in this table, which is not great. So how you can refactor this table into tidy data is you've got your day, which is repeated. You've got your treatment in another column. You've got the concentration in another column. DMSO is obviously usually the vehicle control, so you don't have a concentration here, but don't put 0 because 0 is a number and can skew your data. So it's like it's a missing value, so you need something like NA, which is not applicable, and use that consistently. And then, oh, we've measured the nuclei count. So now we actually have all the information that we need in this table. So messy data can, yes, sometimes be more convenient because it often matches the way the data was collected, stored, or reported. A lot of software creates messy data as an output. It's a direct match to the acquisition format. It is more human readable, yes, that is a fact that I cannot deny. And you've seen this table is much bigger than this table. If you have very, very, very large tables, then there's also an aspect of performance there. And sometimes software compatibility, sometimes software expects messy data. What is the benefit of tidy data? You have consistency for systematic analysis. Every data set follows the same structure, which is not a bad thing. Compatibility works seamlessly with tidyverse tools. This is in R, but also applicable to Python. It's much easier to analyze. You can summarize, you can group, you can use statistical models and apply them very, very easily. You can automate things quickly. Scripts generalize more easily across data sets that are tidy because they don't depend on idiosyncratic formats. Also, reproducibility. It's easier for others and your future self to understand, reuse, and reproduce analyses. And data integration as well. Multiple tidy data sets can easily be joined or bound together without reshaping, and therefore making it easier to combine experiments and get your end numbers up. So lots of positive things. There are no programming tidy data tools like OpenRefine. Your data does remain local, and there are also programming-based tools. So in R, the tidyverse package, and in Python, pandas and numpy packages, if this is something you're doing. Step 3 is methodology and protocols. Literature review. We all need to do it, questions you need to ask yourself. What databases did you search? Which search terms did you use? And you document these. What filters did you use? Why did you include or dismiss certain publications? Have a look at PubMed if there are questionable, problematic publications that then you exclude because there are questions about images, methodologies, other things. Use a reference manager. Whatever your institution provides, or Zotero, for example, is a free-to-use reference manager. Another thing to consider is publishing your protocols. Why should you publish your protocols? Build trust in your work. Enable discovery. Other people can find your protocols and they can use it. They don't have to do it again. Certain protocol databases give you a DOI for your protocol, so it can be cited, and therefore you can expand your publication record. It's an early opportunity for peer review. Somebody might say, oh, that's not the right step. You might want to consider doing something else, or we have found that this works differently. And it's an early record of your novel methodology, software, or innovations. Where can you publish your protocols? Well, protocols.io is quite a popular database. Many journals will publish protocols, and there are dedicated protocol journals like Spring Cold Harbor protocols. And I will provide this presentation later, so when you click on protocol resources, you will get to a bunch of links. For microscopy specifically, what should you report? Well, there are some recommendations for minimal methods reporting. Down here is the link. You need your specimen setup. Which cells are you growing, if it's cells, or which tissue? How is it obtained? How are you labeling your sample? How are you mounting your sample? What kind of consumables are you using? Image acquisition. Which microscope are you using? Objective. Is it lasers? Is it LEDs? Intensities, exposure times, camera gains, averaging, all sorts. But then also the image data itself carries data that needs to be reported. So what I see a lot in publication is images were captured on a Zeiss LSM 800 confocal microscope at 20 times magnification. Try to reproduce that. You will not be able to. It's definitely not enough information to repeat the imaging on the same sample. There are a multitude of 800 confocal microscopes out there. It can be upright, it can be inverted, and there's also a whole host of 20 times magnification objectives. What you would rather want is something like this. You have much more information on what was used. You know exactly, oh, this is a diode laser with 100 milliwatts output set to 10%. Great. You can go, if you have the same microscope, put the same parameters in. If you have a different microscope, you can at least have an idea of where to put your parameters. And yeah, this is much more comprehensive and much more valuable for anybody who was trying to do something similar. Documentation in general. Who is it for? For the boss person, but also for the whole group you're working in. It helps you track what you're doing. It gives a point of reference for your future you and everybody else's future self. It alleviates mental load. You don't have to remember everything. If you've got it written down, it's right there. And you can always get back to it. And you know, was it 5 or was it 0.5? I'm not quite sure. If you've got it written down, you can check it. It acts as a history of change. So it helps you identify where you might have taken a wrong turn. And also consider the bus factor. What is the minimum of number of team members that have to suddenly disappear from a project before the project stalls due to lack of knowledgeable or competent personnel? So yeah, any key member of your team becomes unavailable, What do you need to know to continue their work or to at least keep things steady? Do you know where their work is stored? What about their data? Do you know where their procedures and protocols are stored? How about their research contract, data custodianship details? All of these things are really important. Where to get started with documentation? Maybe there is institutional access to an electronic notebook. Maybe there isn't. A text or Word file stored in the same place as your data. Just a little reminder of the README file in your project. On and offboarding colleagues is an opportunity for documenting. I would say the offboarding is a very late opportunity for documenting, but it's kind of your last chance, really. And it can be tricky to know how much detail to include in protocols and documentations. We all have biases and make assumptions based on our own experience, which is dangerous. Some assumptions you make are maybe not known to a new person in the lab. So it's easy to forget what it is like when you're learning and how much information and knowledge you had as a new member to the team. So onboarding examples, for example, is what are expected contract and work hours? Does the research group post papers as preprints? What technologies are generally used in the group? What analysis tools or methodologies are usually used in the group? How does hardware hire or booking work? How is authorship and author order decided when submitting papers? Offboarding examples is, for example, is a copy of the data being stored at the institute? And where? Is there any colleague within the research group or supervisor that currently also has access to the data? Is this the raw data? Is it process data and final data? Or just one of these? Which is which? What is the retention period of the data? And so on and so forth. So, what to document? Well, your data management plan should have some information there in what you are documenting and how. Did you pre-register your study? This is more for PIs, but if so, where? So you can document the justifications for the decisions made in pre-registration. Did you write a protocol? Is it publicly available? Is it just available within the group? How will you handle missing data? Missing data could be actually missing data, or for example, the concentration in the DMSO column, which is not applicable. When will you stop collecting data? Oh, when the statistical significance is below 0.005? Not a great point. What did not go well in the study? Reasoning for chosen sample sizes, and what settings was your equipment in? What brand and model was that machine that produced the output? All these things are things you should document. Following on from that is you need to consider physical testing and quality assurance. Consider the consumables you use. Are they in date? Are they stored and labeled correctly? Oh, I found it at the back of a fridge. I have no idea how old it is. Were samples affected by transportation between buildings, between sites, between cities even? Is the passage number of cells still appropriate? Consider the hardware you use. Is it calibrated? I'm looking at you, pipettes, pH meters, and similar. Is it producing unexpected effects? Is it compressing, resizing, filtering things that it shouldn't be doing? Is it fit for purpose? For example, does it achieve the necessary resolution? And yeah, comparison with controls is useful for testing things. On a microscopy level is autofluorescence, unspecific binding, photobleaching. These are all really important things. If you are working with data, what is the original data source and what are intermediate files? Or if you're working with secondary data, where did you get get the data from? Have you noted the source of where you get the data from? Your source may not be the, who is it? Was there copyright and access limitations on the data? And if it is a version data like Google Maps shots, which version did you use? Another good thing to do is tracking your analysis history. Keep a backup of raw data. So you can always go back to it. It's also good for accountability. Log your changes, record pipelines. This is particularly important for image analysis pipelines. Save intermediate files for quality control. If you just have your input data and you generate numbers and you have nothing intermediate to look at, whether those numbers actually make sense, you're basically just fishing in the dark. On to version control. Analysis pipelines tend to mature like fine wine or cheese. You're always tweaking, you're always changing, and yes, let's consider tracking the versions of your analysis pipeline. You will likely be making a lot of changes as you go. How are you taking note of these changes? What version of software are you using, be it Fiji, be it Amaris, or any other analysis pipeline, even in Excel? Have you noted what version of the libraries you are using are? So this is more relevant to R or Python. Have you noted the name, model, and version number of any hardware you may be using? So this is the PC, the graphics card, your GPU, that kind of hardware you're using, but also cameras, microscopes, MRI machines, any sort of sensors. Version control is keeping track of each change you have made so that if you need to go back to a previous version of your analysis pipeline, you can. It's kind of like an undo button. It's like, oh, I messed up, I'll go back. So for any code, R, Python, Fiji Macro, MATLAB, whatever it is, Git is your version control friend. GitHub is a cloud platform to track and share code, but you can also keep it private so that nobody can get to it. You can use it to track your changes, but also you can use interactive forms. If you're handing a workflow over to somebody else, like Jupyter, this would be Python, Shiny Apps, this would be in R, which combine plain text and code, so form and function, as in you can put instructions in plain text and then the code does what the code is supposed to be doing. On to automation. Use the KISS principle. Keep it simple, stupid, or Sam. Examples are contact me form, batch image analysis through pipelines or scripts, otherwise you click yourself silly. Data cleaning, you want to automate that to have consistency. Data analysis and visualization, save yourself time, keep it consistent, make everything look nice and neat. Data cross-referencing, project trackers, use software or AI to help generate materials and methods sections from scripts and pipelines. So you can do this with metadata from a microscope and get help in extracting the relevant parameters. Definitely automate that. The next principle I want to introduce is the DRY principle. Don't repeat yourself. Let software do the repetitive tasks for you. Yes, avoid inconsistencies because you might just click the button next to the button you're trying to click and you don't notice. It saves time, energy, and certainly mental load. There are no-code solutions. If you're on Mac, there's the Mac OS Automator. On Microsoft Power Automate, spreadsheet macros, yeah, or Python or R, and formulas will help. But also there are workflow systems such as KNIME and Orange Data Mining, which can help you along with this. Coding is a great tool for automation, and you could use shell scripts to run batches or other scripts at once. Last but not least, the final piece, publishing persistent identifiers and preparing for reuse. So everything else leads up to this point. Persistent identifiers, so different from a name, are long-lasting, unique digital references, for example, to a web page, a digital object, a person, or a facility. Something most of us know is a DOI for a publication, so that's typically what it looks like. Kind of a DOI for a person is an ORCID number. This would be my ORCID number. And then RRID stands for Research Resource Identifier. So this could be an identifier for a piece of equipment, a facility within an institution itself, and these will not change in the future. Anyone has found that a catalog number for a supplier might change over time, and then you're hunting for what is the new thing, but these things are persistent and don't change. So to publish or not to publish, the FAIR principles here are findable, persistent identifier, for example, a DOI. You can get DOIs for data sets. Accessible, you need access through a standard protocol, not a very convoluted, difficult thing. Interoperable, save in an open access format. Even if you generate proprietary data, if you can convert it into an open access format, much, much better. Reusable, permissively license your data set or your protocol, your information, so that somebody else can actually reuse it. Fair data is important. So license provides guidance and sets legal obligations on who can reuse the material and for what. Can this be used commercially? Yes or no? No warranties or similar are given. If somebody uses your work as part of their project, are they obligated to also use the same license? So for me, for these slides, yes, I am obligated to use the same license. Do they need to attribute your work? The license that these slides are under, yes, they do. There is a focal plane post here that I have linked, which gives a lot of very accessible information. So I would really recommend you to read that. It doesn't take that long. Where to deposit your data, or further resources, first of all, is you've got your university library, a Center for Open Science, Guide to Licensing, that's a more generic thing, and yes, data deposition. Where to put your data? If you are starting from very beginning, is use fairsharing.org. It's a repository of repositories. So if you have no idea what you're doing, but you're wanting to publish your data, this is where you could go. There's also a +1 repository guidance page. And for imaging data in particular, there are different options that are there for different use cases. Figshare, Zenodo, Bioimage Archive take mostly light microscopy data, although Bioimage Archive takes anything that's an image. Empire is electron microscopy data. IDR is for standard setting data sets. Is there maybe an institutional database? They're not usually great, but that might be there. And there is a image data deposition decision tree linked here that you can look at and decide where you might want your data to go. Why to deposit your data? It supports your publications. You get a DOI for your work. You're aligning with the FAIR principles. If you are working with things that you need ethical approval for, let's say you're working with mouse, rodents, or any other animal data that is not Drosophila or C. elegans, you're helping other people who might not have the time, the money, or the inclination, or the grant that allows it to generate new data like this, to reuse your data, which is much better for the animals involved in the research as well. How? Check out the data deposition recipe in the FAIR cookbook. That's your best place to go to, first of all. There are online code repositories, so GitHub is one of those, Bitbucket is another one. This is how you can share your code if you want to. I use GitHub myself to share all the code I am allowed to share publicly. And again, you can put links to your repositories in publications so people can find your code. Other repositories that I would like to mention is the KNIME Community Hub, the Bioimage Model Zoo, if you want to use or have created any machine learning based segmentation or other methods, but also published cell profiler pipelines. Publishing negative results. Whilst it can be disheartening to get negative results, these results are still beneficial to the wider research community. You worked out something that didn't work, which is important knowledge all in itself. Sharing this means others don't have to go through the same process to reinvent the wheel, and it saves resources, which is not a bad thing. So just a very quick wrap up. What I would like you to do is take away some actionable advice. Don't try and implement it all at the same time if you're only starting out. I have learned these things, as I said, over many, many years. Spread the word. Ask questions. Ask your PI, ask your community, ask your library, your institution, and be part of the research culture change. Together, we can overcome the reproducibility crisis if AI doesn't make it even harder than it already is by faking any papers. But the real papers, hopefully, we can improve. And with that, I am finished. And thank you very much.
Mari, thank you very much for such a wonderful, easy to follow, resourceful, and timely talk. I really enjoyed your talk. Thank you very much. And now Marie will be very happy to answer any questions. Please, if you have any question, write your question in the question box on the screen, please. So while people are thinking and trying to ask questions, so Marie, may I ask a very simple question? So what are the biggest barriers preventing researchers from sharing their image data?
The biggest barrier I found, one, myself when I did it 10 years ago, 12 years ago now, is not having data organized by project, and then you have to piece everything together from different places. And you can only really do that if you have a record of, let's say, I've created a graph of something, but I have kept a record of which files I have analyzed that contributed to that graph. If you don't have that, then you don't know which data you need to upload to a repository. So I find that this is in practice the biggest stumbling block. People analyze data, but they don't necessarily at the end know which data point comes from which imaging set, and therefore they can't upload the data. And it's also, yeah, it takes time getting all of that data together. And by that point, you've written the paper, it's maybe on a preprint server, and you want to move on. You don't want to put a lot of effort into collating everything that you need for upload, because you need all the metadata, as in your sample preparation and something similar. And you might need, oh, sorry, I've lost you. You might need a lot of time to collate all of this, and this is the biggest stumbling block. So if you follow some of this advice that I've given, you're more likely to have everything in the same place, and then it's much easier to upload data.
Okay, great. So we have a question. So from one of our attendees. So we used white light to observe our HeLa cell culture, and then we used a green filter. We noticed that when using the green filter, the image was better than when we using the white light. We couldn't explain the result. Could you please provide an explanation about that?
So you still use the white light as illumination and then putting like a GFP filter?
Okay.
Is that, yeah. It depends on the light source, really,
Okay.
or maybe there was some fluorescence in there that helped. I don't know.
Okay. So there's another question here. So microscopy files often come in format CZI or ND2. For student wanting to ensure that their data is interoperable, what is the recommended open file format we should use to ensure our images can be opened by any software in future?
Yeah, so CZI and ND2 are not too bad because bioformats can open it. So bioformats is a library that can open a lot of proprietary file formats, including CZI and ND2. If you do want to convert it, depending on the size of your data set, OME-TIFF is a very good file format if the data is not hundreds of gigabytes to terabytes. If you are in such a big file size per data set, it's probably OME-ZARR, Z-A-R-R. That is currently, looks like it's being established as a big data or big file format data format that seems to be sticking around. There's a lot of development going on in that area at the moment, but OME-TIFF and OME-ZARR.
Oh, that's great. So there's another question here. What are the current reproducibility limitations in computational terms at the moment?
In computational terms, lack of
At the moment.
documentation.
Yeah, especially with complex pipelines and potentially non-deterministic systems. Yeah.
Yeah, that is, there's a lot of stuff out there. There are a lot of pipelines that are being published at the moment that don't actually work on somebody else's system. It's making sure that your pipeline is working on a varied number of data sets. So obviously you're working with the data that is generated within your community, but if you could work with collaborators or harness data that has been publicly submitted or has been submitted to a public repository and try your pipeline on other data, which is not as easy as it sounds because sometimes you don't have all the metadata that you need for it, but that's what I'm trying to do increasingly. So I'm reviewing papers sometimes with, hey, here's the new tool and we want you to use it and I can't get it to work, which then I give as feedback. But I'm also trying to make sure if I write a pipeline, have somebody else use it, find out what are their problems, why can they not use it, what are the reasons, and try to make sure and try to make it as applicable as possible. But you can't always test everything because you have Windows, Mac, Linux. Does it work on all the platforms? You can't really say. But yeah, work if you can with your IT section in your institution. And if it's really involved, maybe work with any computer science set up a computer sciences collaboration as well.
That's very important. So there is another question here. So you have mentioned the stigma around publishing negative results and the importance of encouraging the publication or reuse of microscopy data. As a PhD student, what steps would you recommend to make negative or inconclusive results fair and valuable the science community?
Have as many controls as you can to make your conclusions as watertight as possible. And even if you struggle to find a journal to publish it, publish as a preprint. The Bioimage Archive, for example, doesn't care whether you are having imaging data that show positive results, as it were, or negative results. It has to be in conjunction with at least a preprint, so with a publication, obviously with the view of publishing it finally in a journal. So the image repositories usually don't care, so it doesn't matter. If it's images, they take images, but in conjunction with a publication, and preprint is your first step. So that's what I would say. But yeah, have as many controls, talk to other people. Which controls are you maybe missing to really make it watertight, to say this actually doesn't work with as much confidence as the controls give you.
Yes, I agree with you. Controls are extremely important.
Yes.
That basically validate your results. And of course gives you a lot of confidence. So use controls, as many controls as possible whenever you run an experiment. So there is another question here. What are the 3 key pieces of software that you would recommend someone new to the field gains experience with?
A reference manager, something that gives you a step into tidy data. If you have any inclination for coding and you're in the imaging community, Python is the way to go. If you have some experience in R, go with R, that's fine. And I quite like the KNIME analytics platform as well. So these are more on the data analysis side. For image analysis, I try to stick to open source software rather than commercial software like Fiji or QPath, depending on the data, the images you have.
Okay, so we got one more question here. What is your favorite or most recommended repository for depositing very large data, for example, more than 5 gigabyte?
Bioimage Archive. If it's not electron microscopy, Bioimage Archive. If it is electron microscopy, it's Empire.
Empire. Okay, great. So regarding, now AI is very popular. Yeah. So if I ask you, how can we ensure that AI models trained in one lab remain reusable and reproducible when applied in another lab with slightly different image conditions? So
you can't.
Something.
It's the simple answer. But if you have somebody that has machine learning skills, you might be able to refine the model. So the bioimage model Zoo provides the models in different file formats, but these are file formats you can then also use and refine if it does a reasonably good job at segmenting, but it's not quite there. If you have somebody in-house who has machine learning skills, you can then use that model as a starting point, provide more training data. So you need your raw data and your ground truth annotated data to then refine the model to make it more applicable to your data.
Oh, thank you very much.
Which sounds very easy, isn't it? But that's the way to go.
Yeah, yeah, it will, develop further, hopefully, in the coming years.
Hopefully, yes.
Hopefully. So I think now it's 10 to 11 on my side. So, and thank you everyone for attending this webinar. And also thank you very much, Marie, for such a wonderful talk.
You're
And
very welcome.
we can continue the conversation online later on if you want. And we welcome suggestions for future topics and speakers to feature in this biochemistry focused webinar series. If you have any idea for a webinar in 2026, we invite you to submit a proposal for an upcoming webinar. You can find more information on the website of biochemistry. So uh, all our upcoming webinars are listed on our website as well. And if you have missed any of the 100+ webinars that we have already run as part of the series, or would like to watch them again, please visit our website or YouTube channel. The recommending and also the recording from today's webinar will also be available to watch within the next couple of weeks. And finally, I want to highlight that it is more important than ever to stay connected and engage with your fellow molecular bioscientists. Join the Biochemical Society community of researchers and specialists and take advantage of key benefits, including discounted registration fee for society courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journals, and more. Visit their website to find out more information. And thanks to all, and goodbye.